the two cells. So we finish mitosis. So that's it. Mitosis is very simple. Now we talk about meiosis or meiosis. It is the formation of gamete. That's the major function. What are gamete? Gametes, they are sperm and eggs. Just pay attention. For sperm, singular and plural has no S. For eggs, plural has S. Okay? So meiosis occurs only in our for men only occurs in the testes. For women occurs in ovaries. So let me give you an overview of the meiosis. It is very similar to mitosis in that you can see that meiosis, well, the overview is that you have meiosis 1 and meiosis 2. So meiosis 1, remember, it is the homologous synapse? Homologous chromosome separate. Oh, I don't want to use the word synapse. Synapse is... Oh, synapse. Oh. So what happened? They call it synapse because they pair up. Okay, homologous chromosome. Okay, I'll just put it here. For meiosis 1, the homologous chromosome pair up. They call it synapse. Synapse is usually we use it in the nervous system. Homologous chromosome pair up. Synapse here means a pair up and then separate. So this is meiosis 1. So meiosis 1 is a separation of the homologous chromosome. So your mother chromosome, your father chromosome, they are separate. What about meiosis 2? Meiosis 2 is the separation of sister chromatids. Meaning that sister chromatids separate. In other words, this is a very important point. Meiosis 2 is basically the same as mitosis. Not the same, very similar. Similar to mitosis. Remember mitosis we just talked about, it is also the separation of chromatids, okay? So meiosis 2 you may treat it as more or less the same as mitosis because it is separation of the chromatids. Okay, let's take a look at meiosis 1. Meiosis 1, as the name suggested, it also has what? Prophase I, Metaphase I, Anaphase I, and Telophase I. At the end of Telophase I, we call it interkinases. Interkinases meaning that it is in between the division of the cell, in between the division of the cell. So you can see that in here, meiosis 1, at the end of meiosis 1, you also have 2 daughter cells, okay? But what's most important is that the number of chromosomes reduced by half. This is the very important point. At the end of meiosis 1, the number of chromosomes reduced by half. Two N become one N. We call 2N diploid. It should be D. It's not very good. Diploid. One N we call it haploid. In prophase I, basically similar things happen. Nuclear membrane disappear, and then the central centrosome replicate and start to move apart, and then yes, you have the chromosome appear. But during prophase I, you have something called crossover. Crossover is like a general name. The scientific name is called recombination. What's that mean? Meaning that the homologous chromosome, you see the homologous, the synapses. Okay, synapses of a homologue, meaning that the homologous chromosome, that means your father chromosome and your mother chromosome, they pair up. So one is father chromosome, one is the mother chromosome, they pair up. They pair up in the center of the cell, and then what happened? They start to touch each other. The chromatids start to touch each other here. And when the chromatids start to rub and touch each other, then the genetic material exchange. And then we call it recombinant. That's how we call it recombination. Recombination is basically exchanging of the genetic material. So this crossover recombination occur during prophase I. What's so important? What's the significance of this one? Remember this: increase genetic diversity. This is one of the advantages of sexual reproduction. When we produce egg or sperm, the individual sperm or individual egg, we already have genetic diversity. We already generate genetic differences. So the sperm cell contains the DNA material that is different from our body cells. The sperm cell contains the genetic material, the DNA sequence. They are different from our body cell because we have this crossover or recombination. In metaphase I, of course, all the chromosomes, they line up in the middle. We call it equatorial plate. Something called independent assortment occur. What is independent assortment? When the chromosome line up in the middle, you can arrange it like this. See, this is one pair of homologous chromosome. This is another pair. So you can see that all the father chromosome, the paternal chromosome, they are on the left side, and then all the maternal chromosome, they are on the right side. So that at the end of meiosis I, one daughter cell will have all the paternal chromosome. The other daughter cell will have all the maternal chromosome. But independent assortment is that you can have a random organization of the homologous chromosome. You may have the paternal chromosome on the left side of one homologous pair, and then for the other homologous pair, the other paternal chromosome can be on the right side. So at the end of the meiosis one daughter cell will have the paternal chromosome of the shorter one, and the longer one will be the maternal chromosome. The other daughter cell will have the opposite. Shorter one will be the maternal chromosome, longer one will be the paternal chromosome. So in this case, we call this genetic. It also increases the genetic diversity. Meaning that the daughter cell will be different from the mother cell. Same as prophase I, the recombination or crossover produces the daughter cell that are different from the mother cell and also different from your body cell. In here, independent assortment also do that. Well, at this point, try to think about this. We have prophase I followed by metaphase I, meaning that the genetic diversity produced by the crossover or the recombination in prophase I will be nested on the independent assortment produced in the metaphase I. So the genetic diversity will amplify. And then what happened after metaphase I? We have anaphase I, which is, well, for anaphase I, I'm not going to talk about in details, but the anaphase I in here is not separation of chromatids. It is the separation of homologous chromosome. So your mother chromosomes and father chromosome, they will separate. The paternal chromosome and maternal chromosome will separate into two daughter cells. And then the telophase, well, not much. Well, the same thing, reforming of the nuclear membrane, and you have cleavage furrow happen, then you have two daughter cells. Again, I want to emphasize that the two daughter cells at the end of meiosis I, they will be haploid. Okay, the number of chromosomes reduced by half. So what's the importance of meiosis I? See, haploid cell from diploid cell, I already told you that. Genetic variation or genetic diversity is the same thing. Genetic diversity or genetic variation, so that they are different from the parental cell or the body cell by crossover. This is what we call a recombination. Independent assortment. Oh, this is not anaphase I. Should be metaphase I. So for those who watch my video, they get the correction, okay? If people just read my slides, they don't see these mistakes. So here, this is prophase I. Meiosis